Okay. Second to last lecture. Woohoo! Almost there. It is going to be the last lecture, yeah. Yes, so the way it's working out, since we took all of immunity and combined it into one day, we're now going to be able to finish a day early. So on Monday, I'm going to have a full day of review, meaning lecture, I will do a review for you guys for the final, and then during lab, our normal lab time, I'll stay in here. So from 12:30 on, and you guys can ask me questions. We can review topics that you're struggling with, anything like that, but the formal review will have ended. Does that make sense? Okay, cool. So that's the, yes. I haven't decided yet. It's most likely going to be a broad review though. Yeah. Yep. Good question. Okay, cool then. Let's get started. So, we learned yesterday about how our body fights infections. And now we're going to learn the flip side of that, of how do bacteria get around our defenses and how do they attack us? How do they internalize in our cells and all that fun stuff? This is actually pretty cool stuff. We're not going to hit nearly enough of it for me because I happen to love this topic, but we're going to hit the bare bones and a couple cool things. So let's jump in. We're talking about something called pathogenesis, which is how disease is created, basically. Disease from infectious organisms. So we have the term parasite, and we've talked about this a little bit in the microbial ecology portion, but this is an organism that's going to benefit by harming another organism. So technically, any bacteria is going to fall into this category of parasite because they are harming us for their gain. However, when it's something that is bacterial, viral, even though a virus is not an organism, or fungal, and it's a parasite, we call it a pathogen. So bacteria, viruses, and fungi that attack us and parasitize us are called pathogens. And an infection is just when this pathogen or parasite enters or begins to grow on a host. So it's a infection if it's you, it's an infestation if it's your environment. So a lot of times we'll talk about little insect critters that can also cause disease, like lice and ticks and all that. That's technically not an infection, it's an infestation. So that, if you've ever heard that term, that's just another one. So you can be infected with something and not have disease symptoms. It just kind of depends on where the battle is between your immune system and the disease. So there's kind of a sliding line there when it comes to what is disease and how bad is the disease. Then there's two different groups of pathogens that we identify. There's primary pathogens. I rarely ever hear the term primary pathogen used. If you're talking about the medical community or even the research community, we tend to call them frank pathogens. And these are ones that can cause disease in healthy hosts, meaning these are rarely ever microbiome or commensal species. They are there, or if they are there, they are going to be causing disease in you. Just it's how they operate. So Shigella flexneri, this is the one that it's very hard genetically to tell it apart from E. coli. They're basically the same species. Shigella is basically a subspecies of E. coli, but we call it something else. But this is an example of one that we don't ever really have as a commensal, as a microbiome organism. It can cause bacillary dysentery, which is not a lot of fun. And then we have our opportunistic pathogens. Honestly, this is where most pathogens tend to fall. Ones where if you are healthy, if you are not immunocompromised, you're not going to get sick from it unless the bacteria can enter and get past your primary barriers. So it's getting through your skin, getting through your mucous membranes, and somehow getting into your body or into your bloodstream. So these opportunistic pathogens are pathogens of opportunity. They will live on you or in you, and then they see their opening and they're like, haha, I'm going to attack now. So with this, things like Pneumocystis jirovecii, that's this guy right here inside the cyst. This causes a life-threatening infection in AIDS patients. So here you have an immunocompromised host, and it's able to take over their bodies in a lot more easy of a manner than it would otherwise. So this would not really hurt any of us in here. But a better one is Staphylococcus aureus. So at least 30% of the population has this on their skin as a commensal organism. But if you get a cut, or if you poke at a pimple or something like that and you break your skin, they can get in and cause disease. And depending on what variant of Staphylococcus aureus you have, it might be antibiotic resistant, because this is one of our major bacteria that's becoming antibiotic resistant to almost all of our last resort drugs. There was recently, at this point, it was like 8-10 years ago. This spreads pretty rapidly through the homeless communities in California. And there was a lawyer who was trying to help one of her clients who was homeless. He won a case. She hugged him, got MRSA from him, and ended up dying because no antibiotics would treat it. So kind of crazy, but we're going to next class get into the whole problem of antibiotic resistance, because it's a pretty scary one and a pretty big deal. So we'll get into that more later, but there's a preview of what's coming. So pathogenicity is an organism's ability to cause disease. How likely is it to cause disease? So this is really broken down into three things. How easily can it cause disease? How easily can it infect you? How severe is the disease that it can cause? That's virulence. And then we also look at the specific genetic makeup of the pathogen to determine how pathogenic it can be, what possibilities it has, what tools in its arsenal to attack us. And then there's, taking this term virulence, we kind of talk about it in its own way, aside from pathogenicity, because this is one that we really care about. It's the measure of the severity of the disease. So something like Ebola virus, very, very virulent. It passes, it also is very infective, but when you get it, the disease is very harsh, and at one point, 90% of the people who got it died. Now it's somewhere between 60 and 70% of the people who get Ebola die. So pretty virulent. But then something like Bacillus cereus, this is found in soil. We have it on us sometimes. It can cause disease, but rarely, very, very rarely. And any disease it does cause is like a little skin infection, and we kind of shrug it off. Or rhinoviruses, these are ones that can cause like common colds. We just kind of feel like crap for a couple days and then we move on with our lives. That's not as virulent as Ebola virus here. All right. Then there's a lot of things bacteria can do to increase their virulence and make them better disseminate through our tissues, get food from our cells better, get that iron from our cells better. And any tool that they use to increase their virulence is called a virulence factor. So if this factor is removed, it has to be able to affect the organism's ability to cause disease. The problem is this line is kind of fuzzy, because some of our virulence factors are innate parts of the bacteria, like lipopolysaccharide in gram-negative bacteria. That was one of the components of their outer membrane. So we're bringing it all back to the beginning now. This is what we call endotoxin. So yes, if we remove LPS, it'll be harder for that thing to be virulent, but it'll also kill the bacteria because it's a crucial component of their biology. So there's some virulence factors that are just added bonuses that you can remove and now it's harmless, but there's some that are inherent to the organism. Like we talked about yesterday, catalase. Catalase is crucial for bacteria to be able to survive the presence of oxygen. So if you've evolved to live in an oxygen-rich environment, you need catalase to survive. But catalase also helps you survive the phagolysosome. So if you remove that virulence factor, the cell might also just die because it can't handle life in its normal environment, not just that it can no longer escape the phagolysosome, things like that. So there's so many virulence factors. We're not going to be able to talk about all of them, and that makes me sad because they're fun. We mainly talked a lot about them in lab. So things like DNase, when we put the bacteria on the DNase plate, that's a virulence factor. It can break through the neutrophil nets that the neutrophils cast out their DNA to trap the bacteria, and they can eat through it. Coagulase, that clots our blood. It creates a barrier between the bacteria and anything, all of our immune cells trying to come in and attack it. So all of those kinds of things are virulence factors, but we'll talk about others as well. So in order for a bacteria, mainly that's what we're going to talk about. If you want to learn about viruses, take biology of viruses or anything like that instead, because microbiology at CSUN is a much more bacteria-focused class. I apologize for that. But so if microbes want to cause disease, they have to reach a few thresholds first. One, they have to be able to enter the host. So they need tools and tricks to get into our bodies. Then they have to evade the immune system. So they have to learn how to go incognito and prevent the immune system from killing them. They have to be able to like escape the phagolysosome or trick white blood cells and say, you don't want to kill me, I'm friendly, that kind of thing. They have to be able to colonize you. So 1 or 2 bacteria on their own can't do too much until they colonize you and start dividing and copying themselves. And then they can reach a point where they cause more intense disease. They have to usually be able to disseminate through the body. So very rarely does an organism gain entry into your body and then immediately say, okay, this is where I wanted to be the whole time. Especially viruses. They like very particular cells, not just, oh, I need to infect a human cell, but they want to infect a human nerve cell or a human liver cell or whatever it might be. So they need a way to spread through the body. They also need to be able to spread through the body to get away from the immune system, to get away from the other bacteria that they're around so they can go find nutrients and not compete with their brothers and sisters. And then they have to damage the host because that's kind of the whole point. They're using us because we are warm, we have lots of water, we're very stable with all of our, environmental factors because we keep everything with a nice homeostasis, and we are full of lots of good treats, particularly things like iron. So they want to damage us to eat us, basically. So first thing that they have to do is they have to find a way to attach to our cells. If they can't attach, they're going to have a hard time doing anything else. Think about bacteria that are in our gut that want to cause disease. Our gut is constantly moving. If you've ever seen a video or been lucky enough to watch a surgery, it is so cool when you cut something open because the intestines are constantly moving around and constricting and pushing around your food, and it looks like something's like writhing underneath the skin in there. It's so fun. But not only is the body constantly moving, we're constantly pushing things through our digestive system. So if they don't find a way to attach, they're going to get flushed out. And then what was the whole point of them getting entry into our body? So they have to have some way of attaching or adhering to your cells. So often we will call this factor that promotes attachment an adhesin. This is different for viruses than bacteria, and we're mainly going to look at bacteria. But basically, they like to use pili. This is also called Fimbriae, or they'll have other types of proteins on their surface that will allow them to attach to our surface proteins. And that helps them gain entry. And we'll learn that there's pretty cool things they can do that can actually trick our cells to take them in, and then they live inside our cells too, where it's an even safer environment because the immune system can't get you there for the most part. All right, so let's talk about the pili here. So this is the main way that they attach. They look like little hair-like projections all over the cell. Either word is correct to use, so if you use either on the exam, you're fine, but just know that they both basically mean the same thing. This is not the same as the sex pilus. It's a separate type of pilus. Pilus is just effectively saying something sticking out from the cell. The example here that you're seeing in the picture is enterohemorrhagic E. coli, or EHEC. This is a really dangerous variant of E. coli because it can cause, firstly, mainly diarrheal disease. But if you're anyway immunocompromised, or if you don't get the infection taken care of early enough, it can progress to HUS, hemolytic uremic syndrome. This is where it gets into your bloodstream and gets into your kidneys and starts shutting down your kidneys. If your kidneys get shut down, you can't live very long. It's filtering out all the bad stuff in our blood. So this is a very dangerous illness to get. A lot of people die when it progresses to this point. And you can see here the little C bacteria on this plate. This came from a stool sample of a patient. The C ones are the EHEC bacteria. And you can see they have pretty apparent beta hemolysis as well. So they need these type 4 pili to be able to attach in the gut and prevent us from flushing them away. Otherwise, they can't cause all these lovely little symptoms to us. So it's in their best interest to attach is the main point here. Then they can, once they've attached, now their job is, okay, subvert the host immune system and somehow get into this yummy bag of goodies that we've got here in front of us. So the one thing that they can do is create toxins. And there's 2 categories of toxins, exotoxins and endotoxins. I'm going to start with endotoxins because there's really only one, and it's lipopolysaccharide, LPS. That was that 6-legged giraffe that we talked about way at the beginning. The actual part of this that the host does not like is the lipid A portion, the part that's embedded in the outer membrane. So this part out here, the core polysaccharide and O antigen, don't really affect us, but the lipid A does. So if our immune system catches a gram-negative bacteria in the bloodstream and destroys it, it's going to release all those cell particles so that our immune system can sample them and create antibodies for them. Well, if we release lipid A, this causes massive shock throughout the body. And shock, septic shock is bad because, well, it's very hard to treat. And it causes widespread, nearly immediate just drop, a tank in your blood pressure. And that is actually what kills you if you ever have your neck broken. It's not that your neck is broken, it's that depending on where you break the neck, you're severing the control of blood pressure. And that immediate drop in blood pressure is so extreme, you die instantly because you can't get the blood where you need it to go. So the idea with septic shock is kind of the same. It causes widespread inflammation. Your blood vessels will all dilate at the same time because the body is saying, this is in our blood, the problem's in our blood vessels, we need to inflame our blood vessels. And as a result, you go into massive shock and you can die pretty quickly with septic shock. So it basically causes our immune system to go hyperactive. So we have this weird balance where we want to kill these cells, but we also have to be careful that if we kill these cells, they don't release something that's going to kill us too. So that's how endotoxin works. Yeah. I learned this a while ago and now it's gone from my brain. I believe it is a form of septic shock. I don't know necessarily that it's purely due to endotoxin or just a massive infection of the bloodstream. So I can't answer that question directly. I'm sorry. Yeah. Toxic shock syndrome is something that women have to deal with, particularly. Anyone can get it, but it's basically when you pack a wound improperly or during your period, if you keep a tampon in for too long, you get a lot of bacterial growth because blood is delicious to organisms. It's got lots of nutrients in it, so, and iron, big iron hogs, these guys, and you end up with the blood or the bacteria getting into the bloodstream and causing shock. The other type, exotoxins. So endo means in. This is a toxin that's built into the structure of the cell. An exotoxin is one that is produced and secreted or released by the cell. So the idea with these exotoxins is we're going to secrete this compound that can kill the host cell or help us disseminate through tissues better, or unlock nutrients somehow, some way like that. So they have to make them, they have to release them. Sometimes they have to not just release them into the environment, but they have special secretion systems that can actually inject them directly into the cytoplasm of the host cell. We'll talk about those too. But your toxins are going to fall into one of these two categories. Now, there's lots of different mechanisms of action of these toxins. We're really only going to talk about one because it's a very, very common toxin group. But just know that a toxin can disrupt the membrane. It can alter the cytoskeleton. There's some bacteria that this is really cool. What they can do is they can hijack our actin in our cytoskeleton and use it to literally like propel themselves through your cell and push themselves into the neighboring cells too with this actin. They can disrupt our protein synthesis or our cell cycle. They can disrupt signal transduction, which is another one we're going to get into a little bit. They can help the cell adhere better. So a lot of the stuff that the cell will, or that the bacteria will inject into our cell, help it to attach to the cell better and maybe even be internalized by our cell. They can inhibit exocytosis, so the cell can't release anything that could potentially harm the bacteria. They can block the trafficking of vesicles, which is important. If a lysosome is coming towards you and you're in a phagosome, you're like, stop, don't bring that any closer. And then they can also work as super antigens. So here's just a couple pictures of them. We aren't really going to get into the details of any of these except for B here, the AB toxins and how they work. So they consist of two subunits, subunit A and subunit B. This is different than the ones we heard earlier with how the cells will commit suicide, basically. This is a different AB toxin system. The A subunit is the toxicity factor. So this is the one that causes the problem. The B subunits are basically delivery systems. They protect A and get A to where it needs to be. So usually there's 5 B subunits and 1 A subunit. And the B subunits arrange in a ring around the A. So I'm going to go back for a second here. This is what I mean here. We've got the 5 B subunits and the A subunit in the middle of it. And then it's going to be delivered into the cell. So we're going to talk about one of these in particular, but a lot of these A toxins will have what we call ADP ribosyl transferase enzymatic activity. They're going to disrupt G proteins, which affects how much CAMP is made, cyclic AMP. So let's get into how that works. It's actually kind of cool. So yeah, this is just breaking it down a little bit more. This is a different view of the AB subunit and their structure in case you're curious. But let's talk about how it works. One example is the cholera toxin. This is one of the better known examples of an AB toxin, and we're going to talk about it because it's a really great model. Many, many, many other AB toxin systems work in the exact same way as this one. So we're using it as the example. There might be slight differences in other cells, but overall they function the same. So for cholera toxin, do you guys know what cholera is? Most of you? It's not common here, thank God, but it's a bacterial disease that causes massive, massive, massive diarrhea. You get it through the fecal oral route, meaning you drink water or eat food that's contaminated with someone else's feces that has cholera in it, or you're drinking water that has cholera in it, usually due to fecal contamination. But the cholera, the Vibrio species, live in brackish water or salt water anyway. They're big parasites of invertebrates in the ocean. But there's one that found a way to attack us. It gets into your gut and it causes literally just like a waterfall of water coming out of your butt. It's not fun. The main reason why people die with cholera is dehydration. They are losing water so fast that we cannot replace it fast enough. We can't replace their nutrients fast enough, and they end up dying of dehydration. So a common method of treatment without drugs is to give them rice water. So you soak water in rice, or rice in water, and then give them the water that you soaked the rice in, because it has starches and things like that that can help affect water absorption. So why does cholera cause massive diarrhea? The AB toxin. So what they do is the bacteria will bind to what are called gangliosides, and these are receptors on our intestinal linings. And when they bind, it's going to cause the toxin that they produce to be taken up by the cell. So the binding of these receptors internalizes the toxin, and now the toxin is inside a nice little vesicle. What it does from there, it gets released, and then it ADP ribosylates a G protein. Why does that matter? Because G proteins are the ones that control the production of cyclic AMP. So what this particular toxin does is it increases the amount of cyclic AMP produced. It increases the activity of adenylyl cyclase, which produces more CAMP. What the CAMP will do is disrupt ion balances. Mainly, it drives chloride ions out of the cell. So now we've got tons of CAMP, and the CAMP is saying take all the chlorine in the cell and just dump it out. Well, that's fine, I guess, except when you learn osmosis, water doesn't follow its own gradient technically. It follows other gradients. So if we move ions, we're going to cause water to move with it. So when we take all of the chloride ions and suddenly dump them out of the cell, the water says, well, I gotta go with it. And so we lose tons of water. All this water rushes into our intestinal lumen, and our intestines say, yikes, that's a lot of water, we can't hold this, and it comes out the back. And pretty much all the massive diarrheal diseases that you get have some type of AB toxin that works in a similar way of disrupting ion channels and leading to massive water loss. Not fun at all. Any questions about that? All right, cool. Now, LPS, we're going to go back to it for just a minute. It is only in gram negatives, because remember, only gram negatives have that outer membrane, and in that outer membrane is LPS. And a big example of one that we see commonly in hospitals is shock because of Neisseria meningitidis. So this is one that causes meningitis. You get it by breathing it in. So little kids will get this infection, because kids get infections all the time, especially ones that deal with the respiratory system. And somehow the bacteria is able to get from the nasopharynx into the bloodstream, where there it causes that sepsis because of the endotoxin. Now, in the case of this bacteria, what it will do is it will cause a massive depletion of our clotting factors. It'll keep the body from producing more, and it will also kind of bind up whatever is available, for lack of a better word. When this happens, we have massive clotting all throughout our capillaries, and that leads to basically mini thromboses everywhere. And they show up as this little petechial rash. Petechial just refers to like redness. So in this case, we've got these little red rash bumps all over, plus a kid who's having meningitis symptoms and shock symptoms, and you can say pretty conclusively that's probably a gram-negative sepsis of some kind. So that's just an example of how LPS can show up symptomatically in the clinical setting. Any questions? This stuff is fun, right? More fun than the genetics, in my opinion. All right, so we need a way to deploy these toxins. We can just release them if we're a bacteria and just say, here you go, find your home. But wouldn't it be better if these proteins that I'm spending a lot of energy and a lot of resources to make, wouldn't it be better if I can take them and strategically deploy them? Rather than just like carpet bombing a whole area, I'm going to send the drone into the tunnel and just destroy the tunnel kind of idea. So there are secretion systems. So these are different protein structures that allow the cell to target where it secretes these exotoxins. There's type 2 secretion systems. These are very similar to the type 4 pilus, the ones that will help cells attach to things. We're not really going to talk about those anymore at this point. Type 3 secretion systems, their evolution, it comes from flagella. So they will have a structure, a base structure very similar to flagella, and they've just modified it to help them inject things into other cells. This one literally looks and acts like a syringe. And it's very common in a lot of gut pathogens. And also Yersinia pestis, the causative agent of plague, has a pretty important type 3 secretion system. And then there's the type 4, which they have a homology, excuse me, to the sex pilus needed for conjugation. And so they just modified that throughout the years to also be an injectable delivery device. So the type 3 secretion system here, this is what it looks like. Very, very complex structure. They can use ATP mostly to power this. Some of them do use proton motive force. It just depends on the species. And this one has to come into contact with the host cell. And a lot of times the point of this one is to either deliver the proteins directly into the bacterial cytoplasm, or it allows uptake of the cell. So right here, this ruffling, where's the bacteria? Right there's the bacteria. This is Shigella. And we can tell that it is Shigella because its type 3 secretion system creates this membrane ruffling around it. It injects these toxins that causes the membrane around of our host cell to ruffle around it, and it's more likely to say, oh, there's something there, let me pull it in. And then it internalizes the Shigella where it can do its worst work. So these are often on pathogenicity islands, meaning they got it through horizontal gene transfer and then said, oh, this is pretty nice, I'm going to keep it. These are common in lots of our gut bacteria. So Salmonella, Yersinia, Shigella, E. coli, things like that. Very, very common structures. And then we have the type 4 secretion system. Oh, sorry, I'll go back. Saw a lot of people go like, oh, when I change that, sorry. We've got time, I can slow down. It is, yep, sorry, this is supposed to say type 3. Yeah, thank you. Sometimes I'm typing madly fast and my fingers slip. Thanks for catching that. So type 4 secretion systems evolved from the conjugation pilus, and it secretes proteins or proteins plus DNA. So this one can't really secrete just anything. It has a very specific affinity for proteins in particular. And this allows secretion directly from the cytoplasm or the periplasm into the host cytoplasm. And this is found in something called Agrobacterium tumefaciens. It causes tumors in plant cells, or Bordetella pertussis, which causes whooping cough. Whooping cough is, it's a problem mainly for babies. So adults get whooping cough all the time and you kind of don't even really notice it. But babies, because their immune system is naive, and on top of it, their muscles in their neck are so naive too, this can be literally deadly to them. So they can get whooping cough and they have such violent coughing spells that they can actually throw their head back so violently and break their necks and die. So this is a big reason why there's that push of, no, you may not kiss my baby, because you can have whooping cough and not even know it and give it to a little baby who can then die from it. So this is why a lot of parents tend to isolate their kids or not let just anyone hug and kiss them, and no touching of the baby's face kind of thing. Very sad. Another type of Bordetella, not Bordetella pertussis, but a different species, also causes kennel cough in dogs. Similar mechanisms. Any other questions? All right, let's keep going. So a lot of those secretion systems also help the bacteria get into our cells. Well, once they're in there, they have to survive in there too. So how do you survive within our cells? How do you evade our immune system? How do you survive within us to begin with? Because we are a hostile environment. As much as we're bags of delicious meat, we are also very dangerous. We've got a big army. So many of the virulence factors are going to actually help these bacteria escape our immune system as well. Not just deploy toxins and destroy our cells, but to actually hide and evade our immune system. We're going to get into this a little bit. There's so many really cool mechanisms that they use, but we can't talk about all of them. It's just not possible. So we're going to start with talking about how does the bacteria know it's in a host, and then how does it evade the host. So there's a couple ways we can do this. You can avoid the immune system extracellularly. So if you're inside a cell, you don't have quite as many threats from the immune system as you do outside, because outside of a cell, you've got white blood cells that are going to come and attack you and gobble you up. When you're inside a cell, what's the only immune thing you really have to worry about too much? If you've infected and you're inside a cell, what's the only immune cell that you have to worry about? The killer T cells, the cytotoxic T cells, the ones that will notice you're inside the cell and kill the cell you're within. So it's less of a threat. You don't have antibodies, you don't have macrophages, you just have those particular white blood cells. So there's a lot of different ways that they can try to evade our immune system extracellularly. One of the main ones is a capsule. This is a very, very thick polysaccharide. So it's a sugar, saccharide. Polysaccharides are just long, repeated sugar units. And sugars are very sticky, like mucus. So the idea is they secrete this gigantic capsule. I mean, like if the cell is this big, sometimes the capsule can be like that big. And this will allow them to adhere to your cells or to your tissues in general. And it also kind of makes them invisible to our immune system, because all we see is just sugar. We don't see any of the proteins on the surface. So we can't identify this is a foreign invader or not, because all I see is sugar. So helps them kind of invade, stick, and hide from the immune system. Species that cause disease that have capsules, if you can find a way to make them not make their capsule, usually they can't cause disease in us. So this is one of the main virulence factors that those cells have. They can also make proteins to competitively bind to the antibodies. They can say, well, I don't want you to bind to me, so I'm going to flood the area with proteins that you can bind to, and they're going to be like red herrings. You're going to go after my diversion instead of me. You can cause the phagocytes to undergo apoptosis. So if you've got one of those white blood cells on your tail, you can inject them with an endotoxin, or with an exotoxin, excuse me, and cause them to kill themselves so they can't eat you. And then you can also alter your surface antigens. So this helps them hide. This is one of the strategies that our gut microbiome uses to evade our immune system, because they don't want to be eaten by us. They want to help us usually. So they will have different surface antigens that our immune system doesn't recognize quite as easily, or our immune system will recognize closer to self than non-self. This is probably the hardest one for the cell to do, because all of these in the end are based off of random mutations that gave you this ability. But altering surface antigens is very difficult to accomplish through random mutation alone. Our immune system has the capability of creating 10 to the 11 different types of antibodies. That's a lot. I don't even know the word for, like, it's beyond a trillion or quadrillion. I think it's a lot. So they can change their surface antigens, but what are the odds that we have another antibody that can catch them still? So this one is the least likely to happen, but if it does, these types of bacteria are the ones that are going to cause persistent infections, because we can't clear them. You won't be able to clear them with your own immune system because they're evading so well. Any questions about that? It's kind of cool. Yeah. Yes. What was it? The mycoplasmas are good with this. I guess, yeah, it is. Yeah. So things like MAC, things like tuberculosis, that kind of leprosy, those ones tend to do this a lot too. But you can also get inside a cell and have to survive the host defense inside the cell. And a lot of the bacteria that can do this are what we call intracellular pathogens, meaning they've evolved to where they only cause disease once they are inside our cells. That's their niche. So they have three options to avoid being killed by the phagolysosome. One, they can figure out how to grow in there. They can say, your oxidative stress means nothing to me, ha ha ha, and they just live there. They can prevent the lysosome from fusing with the phagosome. So the phagosome is not a hostile environment. It just takes in the bacteria with some extracellular fluid around it. So that's just kind of nice. But it's the fusion with the lysosome that introduces the oxidative burst and the toxic enzymes and all of that. So if we can hijack the cell using exotoxins and say, hey, stop vesicular traffic, so you can't move your lysosome towards me anymore, then you're not going to be killed. You're just going to stay in a nice little protected cocoon of the phagosome. Or you can escape the phagosome entirely. So you get internalized and then you say jailbreak, and you are like, all right, I'm living inside the cell now, not inside your bubble of death. So we're going to talk about a couple of these. Yes.
So would that be the killer T cells are avoiding?
Yes, sort of. Mainly what these are going to do is avoiding white blood cells. So let me change that. T cells are white blood cells. This is avoiding the macrophage or the phagocyte. So the white blood cells that gobble up other bacteria. These cells can still alert the T cells and still be killed by a T cell. But there's also, they have mechanisms on top of this that help them hide from the T cells too. So this is more directly not being killed by a macrophage or a phagocyte. Good question. Yeah. So here's a cool mechanism. This is used by Shigella, and it's also used by Listeria monocytogenes, which it can live and grow in colder temperatures and at our body temperature. So this is the one where you can get it from eating bad deli meat, or it can be on your crops, and it's particularly dangerous for pregnant women. It can cause miscarriages because it can also get across the placenta and kill the baby. So that's why you're not supposed to eat unpasteurized cheeses and deli meats and things like that when you're pregnant. So first thing that the bacteria does is it has to associate with the phagocyte. It has to attach to the phagocyte. Then it's going to inject the proteins, the exotoxins that allow it to be taken up. But rather than blocking the fusion of the phagosome or the lysosome, they just escape the phagosome. They say, this thing is coming to kill me. I've got 30 seconds to get out of this thing before it fuses and I die. So they do. And once they do that, they can actually hijack our cytoskeleton. So they hijack the actin in our cells, and they actually use it to push themselves. It's like a propulsion system. And they will move across the cell and actually use that propulsion system to break into neighboring cells as well. And I just realized this phagocyte here is a typo. I'm sorry about that. Usually this is happening in intestinal cells. So I apologize for that. The phagocyte there, change it to usually gut cells. Because then one of the things that they can do is they can just proliferate through our intestinal cells that way, or they can then move into the other side of our intestinal tract, so not the lumen side where all of our food is, but the body side, and get into our macrophages there and in our bloodstream, and then travel anywhere in the body they want. Once they get into the macrophages can get into blood, they can get into lymph nodes, they can get anywhere, because those are highways to every part of your body. So what type of immunity or immune cell would be required to clear an infection of a bacteria that's escaped the phagosome at this point? So they've done step one here of I've survived the phagosome, I've survived the macrophage. What immune cell can still come in and kill them? Killer T cells, yeah. So not our humoral immunity, our cell immunity. The T cells can come in and still destroy either the intestinal cell or the macrophage that they're in. Now, if the killer T cell wanted to kill our intestinal cell, which MHC class would it be targeting? Class 1 or class 2? What? So which is the one that is associated with any cell? I want to hear I think. I'm hearing a lot of I thinks. So let's see. I'm going to pull it up here really quick.
MHC1.
MHC1, you looked it up?
Yeah.
Yeah. I was going to pull it up for you guys, but yeah, it is MHC class 1. I heard enough people with confidence say it. So MHC class 1 is on any cell. Class 2 is only on the phagocytic cells. So the intestinal cells, they would display antigens on MHC class 1, and that would signal the cytotoxic T cells to kill them. If it's in a macrophage, it's MHC class 2 that would display those antigens and say, I'm infected, kill me. Yep. Any questions about that? That's kind of like a bonus review question for you guys. Alrighty. So there's some other strategies that cells can use. So you can use the type 3 secretion system to enter the phagocyte, and basically, once again, instead of traveling through the cell, it does this other portion here where it gets into our intestinal cells, gets into a macrophage, and that macrophage can then travel wherever it wants in the body. The key is dissemination works best if you get into the blood or the lymph, because those are the superhighways to the rest of the body. If you're just staying in a type of tissue other than blood or lymph, you're not going to get anywhere fast. You're just going to be stuck in that tissue. So if you need to disseminate anywhere, get into the blood. And the best way to do that is a macrophage, because they can get into all of those different types of fluids. All right. Another way that we can avoid the immune system is mimicry. This is very common in the environment. learn about it a lot as kids with like the butterflies and the insects looking like other things. But bacteria can do that too. It's usually molecular mimicry though. We're copying the antigens on our surfaces. So we can make the surface antigens look like the host. We can interfere with the MHC protein. So it's saying, I don't want you to present my antigens at all. So I'm going to prevent you from putting my head on a pike and putting it on your wall. I'm going to stop you from doing that. I'm going to keep my head this time. We can actually change, or bacteria can change the cytokine profiles as well. So we didn't talk about this too much with the immune system, but we release different cytokines that tell our immune system how to behave. We have some cytokines that tell the immune system to calm down. We have some cytokines that say increase inflammation and cause a problem because we're being invaded and things like that. So IL-1 is, interleukin-1 is one of those that increases inflammation and increases our immune response. So some of the bacteria can actually cause our body to secrete interleukin-10 instead. This is one of those that says, shh, nothing to see here. Don't cause inflammation, calm down. And it kind of allows them to evade our immune system because now we're not recruiting white blood cells or anything like that to come and attack them. This is one that's done by Yersinia pestis that causes the plague. We can block the CD8 receptors on the cytotoxic T cells. So we can say we're inside a cell, but I don't want you to kill me. So I can actually block the receptor from being able to attach to this cell, notice that I'm infected, and kill me. And the last one here, we can disrupt ubiquitination. So ubiquitin is a protein tag that we put on proteins destined for degradation. So we'll often do this a lot with bacterial proteins to create antigens to display. So some pathogens don't allow their proteins to be tagged. So if you can't tag them, you can't degrade them, and you can't display them. So therefore, you're evading the immune system still. Any questions about that? Cool, that's it. Any questions in general about this stuff? Yeah. How does Botox? Like a bacteria.
I wouldn't assume that it changes cytokine profile.